Good afternoon, dear students. Today we are back with an excellent topic of pentose phosphate pathway. We have seen the process of glycolysis. We have seen the EMP pathway and we have seen the ED pathway, and now we'll try to comment something regarding this particular pathway. That is what is pentose phosphate pathway, or it is also called as HMP shunt. Pentose phosphate pathway is also called as hexose monophosphate pathway, HMP shunt or phosphogluconate pathway. लक्षात घ्या, आता थर्ड इयरचं वर्ष आहे आणि स्पेसिफिकली एक्झाम मध्ये असे प्रश्न विचारले जाऊ शकतात. Based on that, this slide is said to be very much important because it contains various elements. म्हणजे नुसताच आपण pentose phosphate or PPP pathway म्हणतात त्याला, पण pentose phosphate pathwayच्या ऐवजी it can be asked like hexose monophosphate pathway, HMP shunt or phosphogluconate pathway. It is an alternative route for metabolism of glucose. It is more complex pathway than glycolysis. It is an anabolic in nature. It takes place in the cytosol. Glycolysis जो आपण pathway जो बघितला होता, the glucose to pyruvate चा conversion, pyruvate to TCA cycle funneling, and then it will go in development of electron transport chain, and तिथून आपल्याला high energy molecules like ATP will be generated. हा सगळा part आहे, पण this is another pathway by which glucose can be degraded into this particular product. Tissues such as liver, adipose tissue, adrenal gland, erythrocytes, testes, and lacting mammary glands are elective in HMP shunt. It concerns with the biosynthesis of NADPH and pentoses. Now, what is the biological significance? Importance काय आहे या particular pathway चा? NADPH is used in the synthesis of certain amino acids involving enzymes, glucose dehydrogenase. It is used for the biosynthesis of fatty acids and sterols. The NADPH keeps the glutathione of RBC in reduced state to preserve the integrity of RBC membranes. The NADP keeps the ferrous ions of hemoglobin in the reduced state. The process of photosynthesis requires NADPH. It requires for detoxification of drugs. म्हणजे हा pathway नुसता ग्लुकोज मेटाबॉलिझम साठी वापरला जाणार नाही, तर बाकी सगळ्या ॲबिलिटीज या पर्टिक्युलर पाथवे मुळे आपल्याला मिळू शकतात. So what is the importance of pentoses? Pentoses and the derivatives are used for synthesis of nucleic acids, DNA, RNA, and many nucleotides. When an organism growing on the pentose phosphate pathway or the pentose sugar, this pathway is used to produce carbohydrates for cell wall synthesis. The sequence of reaction of HMP shunt is divided into two phases: oxidative phase and non-oxidative phase. Oxidative phase and non-oxidative म्हणजे काय की जिथं ऑक्सिजन मॉलिक्युल्स आर रिक्वायर्ड आहे फॉर द पर्टिक्युलर मेटाबॉलिझम ऑफ द पर्टिक्युलर शुगर, तेव्हा आपण ती ऑक्सिडेटिव्ह फेज म्हणावी, आणि ज्या ठिकाणी ऑक्सिजनची गरज नाही but the reaction would occur for degradation of that complex molecule of glucose into simple form of energy, इथं आपल्याला इथे ॲबिलिटी मिळते. Glucose 6-phosphate dehydrogenase is an NADP dependent enzyme that converts glucose 6-phosphate into 6-phosphoglucanolactone. It hydrolyzes to form 6-phosphogluconate and then undergoes decarboxylation from ribose 5-phosphate. हा particular pathway आहे जो easily understand होण्यासारखा हा particular pathway. तर या पाथवे मध्ये जर आपण बघितलं तर ग्लुकोज 6-फॉस्फेट 6 कार्बन मॉलिक्यूल आहे. ग्लुकोज 6-फॉस्फेट डिहायड्रोजिनेज जे एन्झाइम आहे ते कन्वर्ट करणार ग्लुकोज 6-फॉस्फेट इनटू 6-फॉस्फोग्लुकॅनोलॅक्टोन. करेक्ट, फक्त कन्वर्जन आहे हे लक्षात घ्या. इथे काय होणार आहे, इथे 6 NADP कन्वर्टेड इनटू 6 NADPH प्लस H. ओके, NADPH फॉर्मेशन इथे होतं आणि 6 कार्बन मॉलिक्यूल्स जे आहेत ती 6 कार्बन मॉलिक्यूल्स आर रिट्राइव्ड ॲज इट इज, त्याच्यामध्ये चेंजेस होणार नाही. 6-phosphoglucanolactone इज नाउ कन्वर्टेड टू 6-phosphogluconate. परत कार्बन मॉलिक्यूल किंवा कार्बनचं जे स्ट्रक्चर आहे ते डिस्टर्ब झालेलं नाहीये. इट इज जस्ट बीन ट्रान्सफर्ड विथ हेल्प ऑफ एन एन्झाइम 6-phosphoglucanolactone hydrolase. 6H2 molecules are combined and 6-phosphogluconate is being generated. This 6-phosphogluconate is now converted to ribulose 5-phosphate with the help of an enzyme called as phosphogluconate dehydrogenase. Another 6 NADP is converted to 6 NADH and plus H plus and CO2 will be released. हा pathway अत्यंत सिंपल असा pathway for degradation of sugar molecules. पण हा पाथवे सगळ्याच ऑर्गॅनिझम्स मध्ये वर्कआउट होईल किंवा सगळ्याच बॉडीच्या पार्ट्स मध्ये वर्कआउट होईल असा भाग नाही. सो बेअर इन माइंड दिस पाथवे विल बी व्हेरी मच स्पेसिफिक. ओके, आता हा जो काही पार्ट होता हा सगळा ऑक्सिडेटिव्ह पार्ट. ओके, नाउ विल कम टू द नॉन ऑक्सिडेटिव्ह पार्ट. द एन्झाइम एपिमरेज कन्वर्ट्स राइबुलोज 5-phosphate into cellulose 5-phosphate. The enzyme Ketoisomerase converts ribulose 5-phosphate into ribose 5-phosphate. The enzyme transketolase catalyzes the transfer of two carbon moieties from xylulose 5-phosphate and ribose 5-phosphate to give a glycerol 3-phosphate and pseudoheptulose 7-phosphate. Transketolase is dependent upon enzyme thymine pyrophosphate EPP plus Ng2 plus ions. The overall reaction may be pleasant 6 glucose 6-phosphate plus 12 NADP plus 6 H2 is 5 glucose 6-phosphate plus 12 NADPH 12 H2 and 6 H2. See, non-oxidative phase मध्ये काय होत आहे 2 मिनिटात आपण बघूया. रायबोलोज 5-phosphate रायबोलोज 5 आणि xylulose 5-phosphate होईल किंवा रायबोज 5-phosphate तयार होत आहे 5 कार्बन atoms मध्ये. Then it may be converted pseudoheptulose 7-phosphate किंवा glycerol 3-phosphate. This glycerol 3-phosphate either when they have a fructose 6-phosphate or erythrose 4-phosphate will be converted with the help of these particular enzymes. Xylulose 5-phosphate gets converted into 6 glucose 6-phosphate, fructose 6-phosphate, glycerol 3-phosphate, and this fructose 6-phosphate as well. म्हणजे degradation of the sugar had taken place into two various different parts. One part was purely an oxidative phase, by which in the presence of oxygen NADH molecules are being generated, 6 and 6 12 NADH molecules, and this is the non-oxidative phase where these particular molecules are being utilized for the degradation of a particular sugar. Regulation. आता हे रेग्युलेट कसं होतं? The regulatory enzymes of HMP shunt pathways are glucose 6-phosphate dehydrogenase and 6-phosphogluconate dehydrogenase. आता प्रॉब्लेम असा आहे की रेग्युलेशन कशासाठी करावं? The regulation has to be done in order to preserve or in order to save the energy of particular cell or cell type. सगळीकडे हा pathway applicable नाही, मी आताच तुम्हाला सांगितलं. And therefore this pathway will be helpful, or this pathway will have an way by which it will degrade sugar in a very specific way. Easily availability नाही, कारण इथं energy खूप लागणार आहे. 6 NADH molecules are highly what we can say high level of molecules which are being needed for this particular path. The synthesis of both enzymes are induced by insulin. The entry of glucose 6-phosphate into the PPP is controlled by cellular concentrate of NADPH. मी जसं म्हटलं तुम्हाला NADPH ची जेव्हा आवश्यकता असणार आहे, एनर्जीची जेव्हा आवश्यकता आहे, अशा वेळेसच this pathway will set to be a very active pathway. Otherwise हा pathway active होणार नाही. So the oxidative phase is controlled by NADPH and the non-oxidative phase is controlled by pentoses. Pentose sugar degradation non-oxidative phase मध्ये होतं and high energy molecules generation हे oxidative phase मध्ये होणार आहे. Correct? So this is a very short pathway which is generated into specific organs of the human body as well as into inside the cell as well. Correct? For the degradation of glucose. Fine. So that's all for today. Next time we'll have some another pathway to work on. Thank you.